So welcome back, everyone. So we have the second session today on the next generation sequencing for clinical applications. And my talk today is not as scientific as the one yesterday, but more a recapitulation of what we learned yesterday and what we will see today, how to get from the raw data to the final list of potentially causal variants. And we will start with a little introduction of the whole topic, and especially what my group is also interested in, is to get from the original genotype to the final disease phenotype and to correlate these two. And obviously there is a lot more than simply our germline genome. There is this whole set of complicated mechanisms that organizes the chromatin, that opens the chromatin, closes the chromatin, activates sites, and so on. So simply knowing the germline genome, we have learned, is not enough. We have to even know our microbiome to get a full view of all diseases. And we have somatic mutations that are not in the germline, especially in cancer, but also in other diseases. They might play a role. So all of this has to be taken into consideration in the long run, also in the clinics. For that, there's many techniques, but the beauty of next generation sequencing is that it's actually covering a large part of all these different things, from the germline genome to the somatic genome to the transcriptome, and then also the epigenome and the conformation of the chromatin. So except for proteins, which can only be studied by other techniques like mass spec, we can look at a large part of the regulation of the chromatin and the mutations. And in this course, we look at a very specific part. We look at the sequencing of the coding genome. In the future, we will probably extend that to the whole genome, although these additional 98% hold a lot of problems for us. Especially, we do not exactly know where all the regulatory mutations are and which ones are regulatory, changing the regulome. In, I don't know what near future, but maybe in the next 10 years, we will also see in the clinics more and more these kind of DNA methylation sequencing, open chromatin sequencing, or protein-DNA interaction. And what's already used a lot, I guess, in clinical research at least, is transcriptome sequencing. And in my group, we have studied a pretty large number of diseases, from rare pediatric genetic disorders. We studied in collaboration with the Estival group, Xavier Estival, who will talk tomorrow, metabolic disorders like cystic fibrosis, phenylketonuria. We studied many cancers, and we have started to look into prenatal diagnostics using exome sequencing. And obviously not exome, but more whole genome is the clinical strains of bacteria, antibiotic-resistant bacteria. The amount of data you get from one bacteria is actually less than an exome, so it's quite nice to work with that. So for the people who may not have a good background in Illumina sequencing technology, I give a single technology slide, how this whole thing works. The idea is that you have your big genome, you sample from a person about 1 million cells, and the DNA from all these 1 million cells you break into small fragments, let's say 200 to 500 base pairs, something like that. So what you see is not the genome of a single cell, but many cells. In the future, this might change. At the moment, we still do sequencing of about 1 million cells per sample. These small fragments you stick on a glass plate. Originally, this was kind of random on this glass plate. The latest generation, they have little holes so that it's ordered. And then you amplify each fragment so that it's a bunch. I don't exactly know how many, but it should be enough that a camera can actually detect them. Then each of these fragments is a single strand, and the idea of Illumina sequencing is that you synthesize the second strand one step by step. There's a little terminator which after each cycle is removed so that the next one can be incorporated. And every time you incorporate a new nucleotide, it's excited by a laser, so it starts to glow in a specific color, and the camera is recording it. So when you look at the whole glass plate, for every fragment, you see in the first cycle you see the first nucleotide incorporated, a T for instance. Then in the next cycle you see the second, another T, and then comes an A. So following over time through these steps, you can on each position get a read. And then on another position, like this one, you get another sequence. So that is the whole idea of sequencing by synthesis. By synthesizing the fragment, you can record every time there is something incorporated. And that can be 1 billion in one single run, so 1 billion fragments, a huge number. And that makes it so useful for exome sequencing of many people in a single run. The exome enrichment is a second important protocol we use for exome sequencing. In this step, from having the whole genome and fragments from the whole genome, we want to enrich into the part that is most interesting for clinical diagnostics, at least what we thought is the most interesting part. The idea is very simple. You have these oligos, which are reverse complement of the sequence you're interested in. So for every part of the exome, the 2% of the genome we're interested in, we have complementary oligos, which are about 100 to 120 base pairs, depending on which technology you use, Agilent or Nimbalgen. They have slightly different length of oligos. So these oligos bind to their complement sequence of your sample, and they have these little biotinylated baits, which you can bind to these magnetic beads, and then you can pull them out with a magnet. So in the end, you pretty much label the sequence you want, and then you pull it out by a magnet, and you get about an enrichment of 80 to 90%. There are still 10% of sequences you don't want, but that cannot be avoided. Unfortunately, also some sequences, about 5% for Agilent and Nimbalgen, do not enrich, although we want them, we don't see them. So, or they are at least drastically underrepresented. So that could be one of the causes why sometimes we do not find the causal variant, is maybe in these regions that do not amplify well. So, well, following that, you pretty much sequence what you pulled out in the standard way, like in whole genome, and then you align it to the genome and find your variants. And here at CRG, we have already sequenced many, many diseases. Here's just a, I think, not complete list, not just in my group, but also a lot in the group of Xavier Esteville. And we could get the price down by doing mass sequencing to less than 500 euros per sample. So it's about 450 if you sequence many in one lane. So if you multiplex by barcoding the sequences, and we can combine 6 in one lane, that would mean in one flow cell, it would be 8 times 6 human exomes we could do. Oh.
Sorry, is this 500 euros or so per exome or targeted exome?
That is per exome.
Whole exome?
Whole exome, but not including the payment for work. So we work for free. Okay. This is purely the cost of the sequencer, the library preparation kit, and the exome enrichment kit to buy them. If you are a company and you have to add cost for your workers and you have to add, well, you want to earn something, this is more like 2,000 to 3,000 euros you buy it in companies, I would say. It depends. I don't know what BGI at the moment takes. Maybe also 1,500 or so is possible. You already saw this picture yesterday. I think Matthias showed it, which is the overview of what has to be done. We just discussed the technology behind the left part. You need some samples, you need to enrich them, build the library, prepare the library, and sequence. This whole thing is kind of standardized nowadays. If you go to a sequencing center, they do it in large scale with robots, and the real problem at the moment starts on the right side, which is the computational analysis. We can produce much more data than we can analyze right now. And with the latest Illumina HiSeq X generation, this will even get worse. First part is already the storage. It's huge files. Whole genome is worse than exome, but even an exome requires 10 to 20 gigabytes of storage. More important is for clinical samples, they have to be secure because they are private data. And if people hack your data and get a hold of them, this might be a problem. Definitely legal, but also ethically. So we want to have secure storage. We yesterday learned about alignment. I left out the quality control here, but we learned about alignment, variant prediction, and in the final step we used ANOVA for annotation. And today we will see more about the inheritance model and variant prioritization. In a more schematic way, we went through kind of this path here. We had a sample, we used FastQC for quality control, we aligned with BWA. Then we did a lot of reprocessing, like we transformed SEM to BAM and we sorted and so on. And then we did a prediction of SNPs and indels using MPylab and VARScan. We didn't really quality filter our results, we just took what VARScan gave us. This would be another step to do if you want to be very strict, you might remove some of the results. And we did the functional annotation with ANOVA. And again, today we will look at the last part, but this last part is actually quite time consuming. And you will get this talk in the end on a memory stick, I think. Well, everyone get a memory stick with all the talks, all the data, so you can follow that. So that's why I included this slide where I recapitulate all the tools you were using, so you can talk to your bioinformatician to install them for you or to set it up in this way. So again, we used for QC, we used FastQC for the FastQ files, and SEM tools, FlexStat for checking the alignment files. And then so on. Alignments, there's a few tools. And for SEM to BAM conversion, we use SEM tools, and for sorting PCAR. Variant calling, we used the first two tools. GTK, I think, is the better one, but it doesn't work on the Galaxy right now. Annotation, we used the first one, but there's a lot of others. And prioritization, that is the tools we will see today. Ediva is developed for my group here at CRG, and the other three are developed in different countries, I think in Belgium and in the US. I put on here this little link, and you will see that this is very interesting because what I have listed there is actually just a minor part of what is really available. There's this interesting site called Omic Tools, omictools.com, and as you can see, they list 2,769 tools for next generation sequencing. If you go in there, what we are at the moment interested in is genome resequencing. We resequence the genome to find SNPs. It's still 800 tools. And then they list just this website, and I think they're not complete, they list 72 aligners, for instance. Bowtie is one of the ones I mentioned to you. They also have annotation tools, so for instance here, and you can see that there's also quite a lot. I don't know how to scroll here. Well, so that means it's not a problem nowadays to find the tool for some application, but to choose the one that fits best to what you want to do. That's why we gave you a list of the standard tools that many people use. But if you have specific problems like structure variant detection, you might have to look for the best tool that is currently suggested by. I learned that the best way to look for new tools is finding a benchmark study. Not very efficient is changing here. Okay. So in general, there's two categories of these tools. Most of them are on command line. That is, when next generation sequencing started, it was purely computational biologists who worked with that stuff. But pretty quickly, people realized that that's not the best for all people. There's companies like CLC Bio who produce graphical user interfaces, and there is this free graphical user interface from the Galaxy project, which we use today, which is pretty much just a graphics tool to use exactly the same that you can use on the command line. We use a free version on the main Galaxy server, which only allows you to work with relatively small files. If you want to seriously use it, you need a local installation of it. And I would say a bioinformatician with some experience in this tool could set it up in a week or so. So now we go step by step very quickly through the different types of analysis we have done yesterday and will do today. The first thing we did yesterday was look at the FastQ format, and we learned that this first column is the name of the sequence, read, and it's kind of constructed from the position on the flow cell and potentially some barcode. This read has no barcode here, but it could have, and then read 1 or read 2, and the name of your machine. Then you have the sequence itself. Then you have this filler line, which normally is just a plus and empty. And then you have the quality string, which is an ASCII encoding, which Mattia explained yesterday, that each of these stands for a value between 0 and 40. So this is one read, and then the next read, and the next, and then typically an exome has about 60 million of these reads. The first thing we did then was to check if we got some good quality FASTQ file, or in paired end sequencing 2 FASTQ files. And the quality is something between 0 and 40. And you can check that in the later nucleotides toward the end of the 100 base pair read, it falls off. And in this specific case I show here, it quite drastically falls off. So you might consider to cut off your reads at 95 in this case. But in the rest of the read, it's fairly okay. Then in the next step, when we decided that the sequence is good, we want to bring the reads onto the genome, on the reference genome. And as we do resequencing here, we need a reference genome here in bold. Without a reference genome, you would have to use other techniques. You would first have to make a reference by assembling your genome. For most genomes you might be interested in, there is nowadays a reference genome, but it's not the case for all. The alignment in general, there are single end or paired end sequencing. In single end sequencing, which is often used for ChIP-seq, you simply want to place your read where it matches best on the reference. The algorithm is very similar to what BLAST is doing, just much, much faster, what BWA is using. The idea is still the same. Like in BLAST, it's a seed and extend. The idea is that you take little pieces of your read. BLAST typically takes between 6 and 12 nucleotides, and you find all the positions in the genome that look like these. And then you take the next 6 and the next 6. And if you're lucky, then they line up somewhere. This is your seed, and then you extend it to the whole read. So this idea is already 30 years old from BLAST. It's still used like that, but just nowadays much more efficient to do it for like 1 billion reads in just a few hours, typically. In paired end sequencing, there's an additional problem. You place your first read and you place your second read, but then afterwards you want to check that they have the distance between them, which you expect from your sheared fragment size. So if the second read falls on a different chromosome or far away, you know there's something wrong. This fragment cannot be from there, or there was a deletion or insertion or some big event. It could also be an inversion. Then you would see this read doesn't look in the right direction. So it would change. Then we went to the Snip calling. In that case, we used a two-step calling approach. We first used MPylab to calculate on each position the total counts. So MPylab came from the first position of chromosome 1, took it and just counted how many T's it sees, and how many reference nucleotides and how many non-reference nucleotides. And it did that for each position and created a so-called PyLab file. So the PyLab doesn't really decide if there's a SNP or not. It just gives you the numbers. Now, theoretically, a human eye could already go through that and just simply count and say, okay, here this is very clear. 8 times non-reference, this must be a SNP. That's exactly what then we did use VARScan for. VARScan uses an algorithm that kind of like we humans decides if there's enough evidence for a SNP at a specific position, and it doesn't look like just simple noise like this position where only 1 or 2 indicate a SNP, but a lot of them say no. And programs like VARScan also decide if that is a homozygous change in both alleles, or if it's rather a heterozygote. And then it gives you a likelihood that it correctly classified the SNP, which is often in GATK called genotype quality. Most of the tools automatically also do short indels. Short indels is everything up to roughly 10. Nowadays a bit longer. BWA now aligns also up to 50 base pair gaps. So many of the tools now automatically look for these inserted or deleted sequence and give you exactly the same information as for the SNPs. Whatever you get from these tools, never trust it. If you overlap the result from several tools, we did that here in a little benchmarking of our own SNP caller Shore. We overlapped with the most commonly used GATK and MPyLab tools, and the intersection was far from 100%. So actually, maybe 70 to 80% of the SNPs are called by all three tools only. Many are called by then only one tool or by two tools. So that means there is not a perfect agreement between SNP callers, which is because they have different thresholds for quality, for coverage, for all of that. In any way, what we found is for an exome, if you take the high quality intersected region, you found about 80,000 SNPs. So that is the number you have to filter down to a single causal variant. It's quite a lot. If you also look at indels, you get, in addition, another 5 to 10,000 indels. And we will later then look into how we will now filter from this to 1 or 10. Just a few values that we typically use for quality is the known percentage of known. We just normally look into dbSNP. If it's in dbSNP, we call it known. If it's not, we call it unknown. If you take the latest dbSNP, we typically get up to 99% known. So only a very small fraction of the SNPs have never been seen before nowadays. You can also do that against 1,000 genomes or anything else. And this is the transition transversion ratio, which is calculated based on if it's a pyrimidine change. And this is in exomes, typically 2.5. You can also see that indels have not been studied that well yet. If you look into dbSNP and check the indels, there is much less known. This could also be that many of them are just simply false, and indel calling is much less specific than SNP calling. So here we expect actually to have 10–20% false positives. The next step we already started yesterday, and we will see again today in more detail, is the annotation of these 80,000 SNPs plus 5 to 10,000 indels. This is a very important step to be able to then filter these SNPs down to the interesting part. So we already saw yesterday from Mattia also these graphics where we have the variant. Our SNP caller already gives us the information of the genotype, how much coverage was there, and the zygosity, heterozygous, homozygous, so on. The first thing we need is a tool that tells us if, for each SNP, if it's synonymous or non-synonymous, or if it's intergenic, intronic, so not even encoding. For the indels, we are more interested if it's a frame shift or an in-frame deletion. And we also want to find splice variants in the splice donor and acceptor, so the first two nucleotides after the exon and before the next exon. Then the most simple analysis to ask if a SNP is important is the conservation in, for instance, the mammals or invertebrates or in apes. So you can go on different levels, but in any way you want to ask, is this a position that is highly conserved in evolution? Then it's likely more important. Third, you want to, based on the protein structure, you want to predict how damaging a non-synonymous SNP might actually be. There's many tools for that. Third, you want to, depending on your disease, find the population allele frequency range that makes sense. For a very rare disease, you want actually something that is less than 1 in 1,000 in the population. If you have a more common disease like diabetes or so, you might actually go higher to several percent in the population would still be acceptable. Again, there's multiple databases giving you allele frequencies in the population. Then we will also see today there's a lot of resources for additional information on the genes, on the disease, on the pathway where the gene is in. That can help you to go down in the number of potential candidates. Also very important, but not always possible, is expression. You could imagine that only genes that are expressed in the disease tissue are of interest. The problem is that for many disease tissues, you cannot get RNA-seq because the patient cannot give you the sample. So if you have a brain disease, you are very unlikely to get brain tissue. If you measure the expression in blood, it is not useful, in this case, to filter the genes, except you have a disease that is affecting blood. And finally, we hopefully have some information from the clinician about the inheritance pattern of this specific disease. In many cases, we also have linkage studies that are done by array analysis or other ways where we already have some potential regions that might actually harbor the gene. So just a few examples of the websites. The probably most important for us to get the population allele frequencies is the 1000 Genomes Project. The latest release, which will be published very soon, has already more than 2,000 cases, so it's not 1000 genomes anymore, it is much more nowadays. And they also divide it into different populations, from Caucasian to Yoruban or African Americans, European Americans, and so on. Another very useful source is from this NHLBI Exome Sequencing project. They studied 6,400, I think, cardiovascular disease patients, and they provided the frequency of the variants in these 6,400 as these exome variants over. So that's a very deep sequencing experiment with more than 6,000 patients, but it's only exome. So with that, you cannot study whole genomes. We have done something very similar, but for Spain, actually mostly Catalonia, I think. It has, for instance, included 450 patients from the chronic lymphocytic leukemia project, which are from Spain, and then many projects we have done here at the CRG, from obsessive compulsive disorder to Parkinson's disease. So we have a similar server where you can look into Spanish population alleles. We have done that because in the past we had some experience that we found interesting candidates in case control studies, and when we validated them, they turned out to be very frequent here in Spain, but totally absent in the 1000 Genomes project. So we hope that is helpful, and it's publicly available. I thought I have the link here, but I don't. But we will see it later on. Then I told you that we can use conservation and damage potential to actually filter the variants to a much smaller size. The first two are examples of conservation. They simply ask how many of the different species have a similar amino acid at this position. And obviously, the more conserved something is, like here, all S, the more important this position must be functionally, so you don't want a variant there. While a position where every species has something else, it's probably not important for the function of the protein. These tools, they go a step further. They integrate this conservation, but they also integrate information from the 3D protein folding from SwissProt and so on. So they take into account amino acid properties and like alpha and beta and so folding structures, and so secondary structure and tertiary structure. And most importantly, all the active sites we know they can take into account, so if there's an active binding site. In our group, we published this very interesting gene in 2012, and you study actually the gene in the first example from yesterday for familial hypokalemic hypertension. In this case, we originally found only a single variant in one family, but then we looked into many more people with this disease and found in total 14 different sites. And interestingly, every one of them had a different position. Only looking at the 3D structure, we found that they kind of cluster in a specific 3D site of the protein, which is important for binding. And you see it in the kind of this form, or also here in this kind of visualization, that they are on the surface and they are all on the same site. So that means you cannot be sure that it's always the same position. That's why exome sequencing is much more powerful than genotyping a single SNP. And it can be in anywhere, even in different domains. So finally, the annotation should end in some form of table. Our tool, Ediva, gives you a nice Excel table. Other tools present it as a document or online, or ANOVA normally gives you a text file. So in that, you would typically have the information from the VCF file still in there, so the chromosome position and the actual change, and maybe the quality, what the SNP caller gave to that call. But then you get additional information of the function, the actually affected gene, and then if it's synonymous, non-synonymous, and then later on you will see there's amino acid change, damage, allele frequencies, all of that. So with that information from this table, we are now hopefully able to filter down the variance from 80,000 to 1. So in the typical, very simplified version of that, you could do it by hand, taking your Excel file and just filtering. For instance, saying, give me everything that is annotated as non-synonymous or splicing, and kick out the rest. That alone brings you down to about 10,000 variants, plus the indels. So we ignore indels here in this graphic. This is SNP only. Still way too much. The next step, which brings you down by another tenfold, is the population allele frequency. Most SNPs you find are high frequency in the population. If you have a rare disease and you put a threshold of 1% here, you probably go from 1,000 to 1,000 SNPs. So that is the rare non-synonymous SNPs in the patient. It could actually be less. I think this one here was done with a 3% allele frequency. Some people even go lower. They say, my disease is so rare, I want maximally 0.1%. Then you will see only 300 left. In the next step, and that is a bit dangerous, we do not suggest to use that as a hard cutoff, is the damage potential. So you could say if all the different tools like SIFT, Polyfen and so on, they say that has no damage, you kick it out. In some cases, that might be wrong. And actually something that looks rare, not damaging, is unfortunately damaging. More important is that the disease model, this is a very clear filter. If the SNP in your family doesn't segregate as you expect, as dominant or recessive or so on, you can remove it. And finally, if you are lucky and for your tissue you have gene expression available, you can filter more on this level. You can hear this, you can also replace by looking at the genes, looking if what other genes they are interacting with in any sort of gene interaction network like KEC pathways or protein interaction networks. Your hope is that you end up with 1 to 10 SNPs, and you will see today that this is about the case for our 2 diseases we will look at today. And these you want to give as a report back to the clinician.
Sorry, population EF is what?
Population allele frequency. Allele frequency. In how many % of the people you actually see it on average. So in 1,000 genomes or in any other population study. So that is, should be distinguished from the in-sample frequency, where in a heterozygous you expect 50-50. Oh, yeah.
Do you think that it's important, like the speaker said yesterday, that every study has their own control group? Control for population or maybe for methodology, technique?
So if you have the Mendelian disease studies where you look at the family, the most important part is that you have a good population study for your population. If you use 1,000 genomes here, but your population is quite different from the people studied in 1,000 genomes, you might consider something rare, which is SNP, which is not rare in your population. So you will not filter perfectly. The other type of study is complex diseases, where you have multi-genes involved, which are typically done by case control studies. So you take maybe 500 Parkinson's disease patients and then 500 healthy, and you compare them to find the genes that are more often affected in the cases. That is a type of study we do not look at this course because you need much larger numbers, but we are doing these.
For example, for cancer with somatic mutations, would you recommend to have a control sample from a different tissue that unaffected by this cancer from the same person? Or because now that you have so much data available, it seems that it's not important to do your own control.
So for somatic mutations, we expect them to be de novo. So we expect them to not be in the population. So if you are strict, we often just remove, if there's a somatic mutation we predict, but it's actually a population SNP, we expect that it was a false positive. We remove it. Otherwise, we do not need a control in somatic because they should be de novo and random. But then to find driver genes, you have to combine many, many patients and find the mutations that are more frequent, the somatic mutations in affected people. You will look at that on Friday, and Luis and Hannah will introduce that for chronic lymphocytic leukemia, for the leukemia study in Spain. They will show you how we do based on 450 patients. We did so on Thursday you will learn, if you have a single cancer patient, how to pinpoint the causal gene by just comparing to databases. And then on the last day they will quickly introduce how to combine many patients to find novel cancer genes. But that is already high-end, actually, stuff.
Another question, sorry. So you have all notation tools for all these selection criteria, or?
Yes. So these will all be in this table, and they are based on, well, as I said here, these are all the selection criteria we need. And the goal of the annotation is to provide all of them, and also add novel stuff if there's a new tool coming out giving another damage score, for instance. We recently added a new one called CAT. Then you can increase the criteria. But the annotation provides you all the information you need, and then the prioritization uses it to either filter or to sort. In the tool we will be using first, Adiva, we do not filter hard, but we sort simply. So the highest one is the most likely and the lowest, the least likely, based on this information. Which information to include? Yeah, that is different actually in every tool. Which of these different criteria they use and how much they weight each of them, how important they think each of the criteria is. So as I said, the disease model is very important for the final filtering to go down to a really small number of candidates. And the tools we look at today, Adiva, Fengene, PhenoDB, they support the following inheritance models. Typically recessive diseases, where both parents are heterozygote and the kid is homozygous. A type of recessive disease, the compound heterozygote, where the same gene has two different mutations in the parents. The unlucky child got both of them. So the gene is, in both cases, the allele is broken, but it's different variants, and the algorithms need to work a bit different. X-linked, you inherit from a healthy mother, and the child is a boy and is sick. We have autosomal dominant. That is actually the disease we looked at yesterday. We looked at the autosomal dominant inheritance pattern. It's typically heterozygous variants and multiple siblings, and also fathers can be sick, so it segregates in the family in a typical dominant pattern. We have de novo. We heard a lot about this yesterday from Christian. You do not find it at all in the mother and father. It's not always fully clear when this de novo mutation occurred. We also heard yesterday it could already be somatic in the parents. But finding them is relatively easy. You look for things that are really new. It's very similar to cancer somatic mutation finding, where the parents are basically a normal tissue. And finally, you have all these multigenic sporadic diseases, which are not studied in small families, but in large panels of case control with hundreds of patients, or GWAS studies, and you might have multiple genes involved, each just explaining why 1 or 2% of the population. So the cancer risk falls into kind of this category. But some cancer risks, like breast cancer, BRCA1, falls into the Mendelian diseases, so the rare ones. Okay, so which tools do we have for prioritization? We will look at today at our tool from CRG called Adiva. I think that's not the latest screenshot. It might look a bit different now. Matthias would know. Yes. So in Adiva, the biggest advantage is you only need the SNP files, nothing else. You need the SNP file in the case of a trio. You just need 3 VCF files, or we will see today you can also use 1 combined VCF file with parents, father, mother, child in there. You just upload it here, and it will give you both the annotation and the prioritization. PhenoDB is in a way similar. You upload your VCF files, and you get, in the end, an annotation and the most likely causal genes. They require a bit more input of disease. I haven't tested it for a while, but I think they ask you more specifically what type of disease and so on. And then we have FenGene. Again, you need the VCF file, but in this case, you also need to know a specific disease classifier, an HPO term that specifies your disease, and you need a pedigree file which explains the pedigree of your family. So we will also start this one today, but here you already need a bit more knowledge and information. So it's a bit more complicated, but gives also quite good results. Finally, we will see today a fourth tool, which actually does not use inheritance information. They use the information that you have to provide of other genes that are already known for your disease. So they use a list of known genes, and then they compare them to all your candidate genes and look for genes that seem to be similar to known disease genes. And then they sort them and color code them. Here the tool is called Endeavor and has a very long website name. So finally, you hopefully end up with 10 or less candidate genes. And now the investigation starts that nobody can help you with, except for a few web resources that you can check for additional information. Well, obviously most commonly used is PubMed. You can just ask PubMed if it knows this gene name together with your disease, and hopefully, or if you want to publish, hopefully not, you get something. The UCSC Genome Browser, I think, is the most comprehensive tool to actually look at your candidate gene in more detail and get a lot of expression information and even epigenetic information. And they also provide a lot of links to other resources. So I would always start with actually these probably. Another resource that is similar is GeneCards. GeneCards collects pretty much for every gene all links that are available and links to everything from OMIM to protein interaction to browsers. So GeneCards is very good get it all source. And finally, you want to definitely check out OMIM, what is actually known about your gene already in form of in relation to disease. And then what we often do is using protein interaction networks or pathway tools like StringDB, put in your gene and check out what other genes it's interacting with. Maybe you recognize one of them as that is already known. So these are some of the ways to filter your final 10 genes where no program can help you anymore to make the final decision. Here's a picture of the Reactome pathway database, which you might also use to find out if your gene is interacting with known pathways that fit to your disease. And I mentioned OMIM, so in OMIM you can search for any term. For instance, if you search for ataxia, you will get a long list of different ataxias, and then you have to find the one that fits best to your clinical phenotype. And you can check out the description, and you will get a list of potentially already known genes. And also suggestions what are closely related phenotypes. Maybe you misdiagnosed your patient. Good, and very quickly to give you an idea of what cases we study. The first one we started yesterday is this familial hyperkalemic hypertension. It's from southern France. We collaborated with a professor from Nantes, and he was approaching us with 3 brothers from a family who were all affected by this kind of hypertension, which is related to, you can see it here, the positive sodium-ion balance and renal potassium-ion retention. Well, what we knew when we started is that in France, about 10% of the people having this specific disease were affected by mutations in WINK1 and WINK4, which lead to increased activity of the natrium chloride, sodium chloride co-transporter NCC. But that is not much. That means 90% of the people were unknown why they were actually affected. So we used that family. There were at least 5 people affected. I don't remember which of the ones we chose, but typically what we do is we choose the most distant relatives to have more meiosis in between them. So you would maybe choose that one, that, and that one, for instance. Then we did exome sequencing. We did this typical filtering, just a shortcut here. It should be non-synonymous splicing. You have the prioritization by allele frequency and so on. In that case, we also already had a linkage information from Nantes, where they said it's likely on chromosome 5 in a specific region. So that made it a bit easier, because we originally, without linkage, had first 2 genes left, and then one was in the linkage region. And then you validate it by Sanger. I didn't show you the gene here because maybe you want to find it yourself today, instead of just handed it here. So this was the inheritance model, the only one we actually considered, dominant inheritance. If you do not find any candidate, you might want to look into more not so obvious inheritance models which don't fit, but maybe again you misclassified your case somehow. And in the final step, we took, I think in total, 33 families with a similar phenotype, and we exome sequenced them all, and we found then actually that from, it was 38, that 14 of them, 33%, had this gene affected. So this seems to be quite a big player in this specific rare disease. Good. And I showed you this picture before. They had an interesting pattern here, that all the mutations clustered on the surface and were seemingly kind of an interaction site. And we found that this interaction site is actually the same interactor, the sodium chloride cotransporter, which already the other known genes interact to. So interaction data can be very helpful to make sure you found the right gene. The other 2 studies we have on the hard disk, which you can use. I will only show you the OMIM site. The one we will run today is myasthenic syndrome, congenital myasthenic syndrome from the Hospital Val-de-Bron here, from collaboration with Francina Monel. And the other one is with Alphonse Macaya, also from the pediatrics here in Val-de-Bron, is a spinocerebellar ataxia. And you can read the OMIP entries if you're interested in these. It's actually not so easy to find the right one for ataxia. There's probably 35 different classifications, and the same for myasthenic syndrome. So finding the exact right diagnostics is not so easy, and it's actually quite important for us because there might be different genes that we should look for. As the final slide, what was mentioned yesterday, if you are only interested in diagnostics and not in finding novel genes, it is probably much better to look at a small panel, or how Christian called it, a package of genes. And for that, you wouldn't actually have to sequence the exome. You can save a lot of money by, well, here, by just choosing one of the disease targeted sequencing panels. The only difference here is that you do not enrich the whole 20,000 genes, but you enrich a subset. And you can buy from Illumina or from other companies like Agilent and so on. You can get these pre-made panels of genes, but Agilent, Nimble, and Illumina also allow you to make your own. So you go to the website, you put in the sequence of your genes, and they will give you these 200 genes. There's many advantages. You can sequence much more in one lane, so it's much cheaper. Second, you do not have to care about incidental findings in genes that are not important for you, so you don't have to think ethically about the implications if you find out that this person gets Parkinson soon. And in general, the analysis is simpler. So for instance, if you do 200 genes, that's about 100 times smaller file that you get than from exome. So even on Galaxy, you can easily analyze several without getting into space problems. I think that will be the first application that will be very, very broadly used in the hospitals before exome even becomes the norm. Many hospitals will first start with the targeted panels.